Very good afternoon, one and all. I am Arpita, the moderator for today's session from the IGCP group. The topic that we are going to cover today is non hormonal contraception, new developments, and patient preferences. And yes, to cover this topic, we have with us someone very eminent and very known to all of us, Dr. Sujit as sir. It's an opportunity for me and a pleasure for me to welcome sir and to introduce him to everyone. So it will be a very brief one. So has done MBBS, then Diplomat of National Board in Obstetrics and Gynecology, his fellowship in Minimal Access Surgery, diploma in gynae endoscopy. He's a senior consultant in gynae and ops department at PD Hinduja Hospital, Mumbai. He is very known for his podcast channel that is Guy from Gynac, has been creating a buzz on all social media platforms and all women-centric sensitive topics and health concerns and be discussed there. So with this very brief introduction of him, I would now request him to please start with the session for today. Over to you, sir. Sir, kindly unmute.
Thank you, everyone. Good afternoon, Arpita. Good afternoon, everyone who is gathered here from IGCP. So today we'll be talking about non-hormonal contraception. The idea is that we have still unintended pregnancies happening everywhere in the world, in spite of all the contraceptives that we have developed since ages. And contraception is something which is historic. It has seen development right from maybe 100 years AD till current. The ones that we are expecting or the using right now are the hormonal contraception, which include the pills, the rings, the injectables, and the IUDs, which are easily available, and most of them are very effective. But the only concern that comes up is with the hormonal side effects for the patients. We have non-hormonal contraceptions, which include permanent sterilization, the one which gives you almost 100% efficacy. Apart from that, we have copper IUDs, chemical barriers like spermicides, physical barriers like condoms, the traditional family planning methods like withdrawal method or the rhythm method, and individuals who wish to retain long-term fertility but also want contraception for a longer time and which is reversible, or for long acting reversible contraception. The only one, if we have to think about in this heading, is the copper IUD. So, what decides that this is the contraception that one person will opt for? Well, there are very varied influences that can affect the decision, including whatever is available, the cost of contraception, the ease which one can use it, the benefits, the side effects, or the stigmatization around it, or the lack of education. At times, even misconceptions around certain contraceptions, majorly the pills, also can decide whether they want to go ahead with it or not. Also, cultural differences can exist around the contraception decision. For example, I've given you a small example there that a study across several urban African countries observed that communities where women believing in contraception myths, such as the statement, people who use contraceptives end up with health problems, had increased contraceptive use. Whereas, in contrast, the communities where men firmly believed these myths had decreased the utilization. So, this is how cultural differences can also affect the decision of contraception. But why is there a need? Because hormonal contraceptions are there, but they do have systemic side effects in general. So, we need to find out certain non hormonal contraceptions which are deemed as safe and which are more accepted, but there is a large unmet demand around it. In the existing non-hormonal contraception, there is a list. I'll just quickly run through it. We know about the permanent sterilization, like the tubal ligations and the vasectomies. They are permanent, long-lasting, and they work almost 100%. One needs to remember that once done, this can't be reversible or takes surgeries, or even the reversible is hardly effective. So this is one decision that the couples have to make when they are moving ahead with this. Apart from that, we have the intrauterine device, which is like the copper T, which works well, and it can also be considered as a long-acting reversible contraception. We have the copper T, which was first approved by FDA in 1984, which was called as Paragard, and then it undergo a lot of changes regarding the sizes and the winding of the copper around, and now we have the copper T 380A. Overall, copper T is very effective and it's better compared to hormonal contraceptions also, and can be used irrespective of the BMI of the patient. However, when you need to insert or remove, you need a physician which has to be trained in removing or putting the copper T. Chemical barriers can include your spermicides, which can come as gels, films, creams, or suppositories. The traditional one, or the one which we are using even to date, is a non-oxidol 9, which is basically something which disrupts the membrane of the sperm, leading to sperm immobilization and eventually sperm death. The newer thing which has come in is Fexi, which is available. It's not a recent advance. It has been available nowadays. It is basically a chemical barrier which causes buffering of the vaginal environment to maintain an acidic pH, and that is how it acts as a spermicidal. It is available via prescription and only used as a topical contraceptive. Benefit with Fexi is it gives 86% effectivity against preventing pregnancies. Physical barriers, as we all know, are the condoms, the diaphragm, the sponges, and the cervical caps. This is something which is only the only contraceptive which is effective in preventing sexually transmitted infections, and it is one of the most commonly available contraceptives that we hear about and that is used. Traditional family planning methods includes your coitus interruptus, your rhythm method, your Billings method, and the basal body temperature method that we all know about. It's least effective, but then this is something which is being followed, and approximately still 19 million women rely on the traditional contraceptive methods. So now we come to the major chunk of the discussion, that is the currently newer developing contraceptives, which are non-hormonal. So locally acting, these are divided under basic headings. The first heading that we talk about is locally acting microbiomes and spermicidals. So we know that spermicidal exists, like non-oxidol 9, which is an effective contraceptive agent, and also we need to now think about what else are available. Fexi has capitalized on the existing market currently available by delivering lactic acid, citric acid, and potassium bitartrate as an intravaginal gel, and that leads to spermiostatic and spermicidal activity. Additionally, some antimicrobials have also shown in preventing sexually transmitted infections, making them especially of interest in multi-purpose prevention team, where Fexi is also showing effective in preventing HIV also, along with its contraceptives benefit. So much like Fexi, we have something which is called as Ovaprene. Ovaprene is developed, which is a monthly silicone intravaginal ring with knitted polymer mesh fitted, filling the middle of the ring to provide the physical sperm barrier while delivering ferrous gluconate and ascorbic acid to reduce sperm motility. So it's basically like a ring with a mesh inside. The mesh will prevent the sperm to go in, whereas the ring has the ferrous gluconate and ascorbic acid, which affects the sperm motility. It is under trials and soon will be available for dispersal. Upon approval, Ovaprene would be the first reversible yet long-acting user-controlled non-hormonal contraceptive. And it is also said that the concerns around it is exist that it can be leading to alteration of the vaginal microbiome, which can be somewhere surrounding bacterial vaginosis. Therefore, one needs to be aware about this aspect whenever using a Ovaprene. Unipron is another antimicrobial vaginal agent in development. This utilizes citric acid to maintain the acidic vaginal environment, and it is shown to be safe and efficacious in non-human primates. Additional studies also show that Unipron and citric acid has been effective in combating certain STIs also. The polymeric SMA, that is the styrene maleic acid anhydride, is used in intravasal male contraceptive hydrogel called Risog. Risog is something which is doing a lot of talk, especially in India. Risog is reversible inhibition of sperm under guidance, which is basically also modifying the pH around it and leads to inhibition of sperm under guidance. While the driving spermicidal action of SMA is unclear, several mechanisms have been proposed, including lowering the pH, which may be caused by the positively charged precipitates that interact with sperm during ejaculation. This is currently completed trials in India and is marketed as a less invasive alternative to vasectomy. Preclinical animal studies show that Risug may be reversible through dissolution of the polymers, but currently we are marketing as a very famous or very new contraceptive in India, as it causes partial occlusion and the polymer hydrolyzes after injection in the vas deferens. Similar to that, we have a cuproferrocomposite of SMA called Smart Risuk. It's been actively developed and studied for better detectability and contraceptive control. In US, the Risuk technology has been licensed, and development of another intravasal injection of SMA contraceptive is being in study, which is called the Visa gel. So we have the Risuk, the Smart Risuk, and the Vasal gel. Research indicates that nearly one-third of male college students are interested in using the technology, while other had uncertainties, most often due to the novelty and the route of administration. Preliminary work has also begun to utilize the SMA hydrogel as an occlusive female contraceptive through implantation in the fallopian tubes. But around this, what we know about is the Risuk, which is available, the Smart Risuk, and the Vasal gel, which is available, which basically causes development of a plaque kind of a thing in the vas, and that will cause the male contraceptive benefit. Polystyrene sulfonate, that is PSS, has also shown efficacy in phase 1 trial as the active ingredient in female contraceptive gel. Currently approved to treat elevated potassium in plasma, PSS has been shown to inhibit several sperm-related enzymes critical to fertilization, such as acrosin and hyaluronidase. Furthermore, its possesses antimicrobial properties, thus effective against several STIs, including HIV and chlamydia. PSS was formulated into an intravaginal film, but no further studies have been published. So this is one thing which is again under research. A variety of naturally derived antimicrobials have been studied for the contraceptive benefits, though none of them progressed beyond clinical studies. Manganine, in isolated from frog mucosal secretions, and niacin, a product of Lactococcus lactis found in dairy products, are antimicrobial peptides, and they possess both antipathogenic and spermicidal or spermiostatic properties, respectively. Curcumin, or what we know as haldi or turmeric, can significantly reduce sperm motility and function upon exposure to higher concentrations. Its broad antimicrobial benefits and general high tolerability inspire potential for additional therapeutic and contraceptive use. Polymeric nanomesh fibers loaded with glycerol monolaurate or GML found in coconut oil have been also shown in vitro effective against HIV and sperms, and that can also be embedded into lipid membranes and inhibit signal transduction. Lupiol is another contraceptive antimicrobial. Though the mechanism is unclear, lupiol is found in a variety of fruits and vegetables, and its anti-inflammatory benefit have been documented. Lupiol has observed to affect sperm hyperactivation, a stage of enhanced sperm motility necessary for egg fertilization. Though the source of this effect is contested, it is originally believed that lupiol inhibited hyperactivation by binding to ABSD2 and obstructing activation of the sperm calcium channels cat sperm. But we have been not able to replicate that on the research level on a larger scale. So these are few things which are available naturally and can be considered as contraceptives later on in the future with more research which are awaited. Coming to the next aspect is small molecule reproductive protein inhibitor. We know that spermatogenesis is a continuous sequence of cell proliferation and differentiation of the germ cell, which results in the forming of the spermatozoa. Following the onset of puberty, spermatogenesis occurs continuously with millions of sperms which are produced daily. This makes protein relevant to spermatogenesis extremely available for male contraception. And whenever you affect or change the protein, it can lead to fertility, infertility, or contraceptive benefits. Several proteins have been established on this, deciding upon the contraceptive aspect. Testis-specific bromodomain (BRDT) is an epigenetic read of protein expression male germ cells, the BRDT. Inhibition of the BRDT hinders chromatin remodeling, which leads to spermatozoa developing with irregular morphology and motility. The effect of small molecular inhibitor called GQ1 was studied and complete, yet reversible contraception in mice was observed. GQ1 has not been developed yet as it binds off target to transcription regulating bromodomain in protein throughout the body. So it's not just specific to the male germ cells, but it can act throughout the body. Additionally, GQ1 was shown to rapidly clear from the system with administered orally or intravenously to the mice, further limiting its potential by requiring exceeding frequent doses. So within half an hour, if the T half is 1 to 2 hours, it will once you take it, it disappears within the system within like 2 to 4 hours. So it might require frequent administration of doses. The development of GQ1 highlighted the potential of targeting BRDT for contraception, though newly highly selective inhibitors are currently being developed and screened for contraceptive potential. Since we know that GQ1 was something which was affecting the bromodomain throughout the body, we need to have a specific thing which is targeting the male germ cells and have a longer half-life so that this can be effective on a long-term basis. The nuclear retinoic acid receptor, the RARs, are male contraceptive targets because retinoic acid has an important role in several male reproductive processes, including spermatogenic differentiation. Male mice with RAR genetic knockout are completely infertile with no observed side effects. Several small molecule antagonists have been screened for selection against RAR, with recent discovery of YCT529 garnering attention due to its contraceptive success following oral administration in mice. So this is at a molecular level, which is done on the mice, which affects the retinoic acid receptors, which are including in the spermatogenic differentiation. Another thing which is called the HIPK4, the homeodomain interacting protein kinase 4, is expressed in spermatids and is essential for spermatozoa to develop the elongated phenotype. The family of testis-specific serine threokinases, that is the TSSK, are also necessary components for spermatogenic differentiation. Additional protein kinases crucial to sperm formation and maturation have been identified. The specific drug candidates for this are yet to be developed. Many drug candidates that inhibit spermatogenesis through a multitude of protein targets have also been identified. Among that, the oleanic acid or the triterpenoid, commonly found in plants, have been shown to reduce fertility and sperm motility in male rats. The mechanism by which this occurs is currently undetermined, but it is said that it causes disruption of the intercirculatory junctions, resulting in increasing in the blood testis barrier permeability, which can expose immature sperms to toxic agents. So this is how the oleanic acid works. There's another called antioxidant, the beta caryophyllene, which contributes to the spice of the black pepper and clove that we know about. It's also an agonist of the type 2 cannabinoids receptor. These receptors are commonly expressed in immune-related cells during active inflammation, as well as in testicular germ cells. Male rats treated with beta caryophyllene had reduced sperm motility, abnormal sperm morphology, and reduced total sperm count. So this also opens a domain where you can consider any contraceptive benefit. Elongating and the elongated spermatids anchor themselves to Sertoli cells, the somatic cells of testicular epithelium that offer support during spermatogenesis for stability during maturation. When you disrupt this interaction, this can cause into contraceptive benefit. The herb derived tryptanoid binds to placoglobulin, which mediates desmosomal junction between Sertoli cells and between Sertoli cells and germ cells. This binding is believed to inhibit the essential interaction between desmosomal placoglobulin and its partner, the SPM, exclusively found in elongating and elongated spermatids. So, tryptonide may impede nucleocytoplasmic transport, resulting in abnormal sperm morphology. Non-human primates, when we had a study which was done with tryptonide, it was observed that the contraceptive efficacy of over 2 years without any observation side effects. Additionally, sperm morphology and function were restored within weeks of treatment cessation, making tryptonide an especially attractive component of reversible male contraception. So basically, to give you a gist after this slide, is tryptonide is something which binds at junctions between the Sertoli cells and junction between the Sertoli cells and the germ cells. This leads to alteration in the sperm morphology, and that is how it leads to the contraceptive benefit. This acts for around 2 years, and once stopped, the cessation of the action of tryptonide is very fast, so it can act as a very good reversible contraceptive in the future. Non-loneramide analogs such as the adjudin and the H2-gamindazole, which has also been observed to interfere with the Sertoli germ cell junction. That is what we discussed about pellet. The phosphodiesterase, which is found in female reproductive system, also may offer a unique pathway when we think about female contraception and maturation of female oocyte to before the ovulation. So phosphodiesterase 3A located in the oocyte prevents accumulation of cyclic adenosine monophosphate, the CAMP. Inhibition of the PDE3 prevents CAMP degradation, which inhibits meiotic resumption, rendering monthly ovulated cycles as immature or non-fertilizable. So basically, when you affect these channels, the egg which is released is immature and it is not something which can be fertilized. Inhibitors have shown that maintaining the meiotic arrest in vitro, such as commercially available milrinone, is something which is milrinone is anyways used in pulmonary vasodilation. But when you're using this, it can be effective in causing female infertility by rendering the oocytes immature. The third thing is disruption of sperm transit. The sperm protein epin or the epididymal protease inhibitor plays an important role in sperm motility. And if we damage these areas, this will lead to temporary loss of sperm motility by gelatin. So PSA typically reverses this through the semen liquefaction, but the investigative and the highly selective drug compound EP055 irreversibly binds with epin and prevents liquefaction. Intravenous administration of this drug has resulted in reversible contraception in male macaque studies. PSA, sometimes known as KLK3, has also been inhibited through vaginal administration of AEBSF. This safely prevented semen liquefaction in mice, but the compound showed human endocervical cell toxicity. That is why it's not something which is being under study right now. So basically, we are affecting the sperm motility by using different mechanisms which affect the epin, and that prevents the liquefaction and leads to clumping of sperms. Several ion transport mechanisms have been significant effects on sperm motility. What we know about is the sodium potassium ATPase4, the K-spur, the cat spur that we already read about, which are the three specific ion channels. This is where we are targeting, and we are hoping that this will this we have certain medications like the cat spur inhibitor HC056456 has been created. So sperm capacitation preceding ejaculation further relies on activation of soluble adenylyl cyclase forms like the ACL and the ASACLF, which are not found in somatics. So these have aided in the development of highly selective inhibitors with potential for both male and female contraceptive control. So basically, you're targeting the iron levels, and these will cause the either specific targets or which affects the capacitation process, and this is how we are hoping that we achieve contraceptive benefit. Disruption of fertilization and implantation. Although contraceptives targets disrupting oogenesis are extremely limited, non-hormonal targets to disrupt ovulation are more promising. So what we have is the cyclooxygenase, which is what we anyways use for inflammation, and by production of prostaglandins. Both enzymes have been studied as potential targets for non-hormonal female contraception. COX inhibitors such as cereblex and the meloxicam have shown inhibition of ovulation in non-human primates, which showed efficacy as emergency contraception, but not primarily birth control. However, clinical studies are on which revealed that cereblex did not have substantial potential as an emergency contraceptive in humans, though it's effective in non-human primates. In contrast, twice daily administration of BAY06, a prostaglandin antagonist, had resulted in substantial yet reversible decrease in fertility, though additional research has yet to be conducted. The ATP binding acid, the ABCC4, has recently been found to be important prostaglandin transporter and is upregulated in preparation of ovulation. ABCC4 may be another contraceptive target for inhibition of ovulation by reducing effective prostaglandin reflux. The oocyte-specific kinase, the WEE2, the V2, is a key player in the final meiotic resumption and overall oocyte maturation that we were talking about. Preliminary development of V2 selective inhibitors identified with throughout process screening is underway. So we have a handful of fusion proteins, the Juno, the Izumo, the THEM, which have been identified, which are a part of ligand-based protein receptor between the sperm and the egg. So once we think about damaging this receptor between the egg and the sperm, we can achieve contraceptive benefit by targeting this small molecule development complex. Knockout genetic studies have been ascertained proteins specifically responsible for embryonic implantation that will lead to development of female non-hormonal contraceptives. What we have currently is the serum and glucocorticoid inducible kinase, that's the SGK, and the proprotein convertase, that's the PC6, are enzymes crucial for developing endometrial epithelial receptivity to implantation. So if you're targeting the SGK and the PC6, the endometrial epithelial receptivity is affected. Therefore, it might prevent implantations. Interestingly, a decline of SGK is required to enable endothelial receptivity, though higher levels are required to maintain pregnancy. The nona-D-arginine or poly-R is a polypeptide-based receptor of PC6, which has shown to be shown the ability to prevent implantation also. Other known implantation proteins, which are contraceptive targets that include the cytokines LIF6, IL-11. LIF6 binds to uterine epithelial proteins, and this is somewhere we can again target to prevent embryo implantation. Pegylated LIF antagonists have shown to inhibit human embryo attachments. Similarly, IL-11 have been identified as a key cytokine for embryo implantation. Pegylated IL-11 antagonist administered has also been shown to reduce implantation site by altering the formulation of decidual cells. We have the immunocontraception. These are immunomodulation that are done so that the sperm antibodies are produced, which will render fertility issues. Numerous sperm proteins have been identified and characterized. What we have are like PH20, the LDHC4, and the Izumo. LDHC4 is one of the earliest sperm antigens studied for efficacy in non-human primates. The immunodominant B cell epitope of the enzyme was chimerized with stimulating tetanus T20 epitope and used to elicit immunogenic response in baboons. Unfortunately, when we tried to reduce the fertility, only 62% reduction was observed. The sperm surface hyaluronidase PH20, also known as SPAM1, facilitated several critical processes during fertilization, including zonal penetration. It is highly conserved among mammalian models of human contraception, such as rodents, bovines, monkeys, etc. In main guinea pigs, when we thought about immunization, PH20 provided a complete and reversible loss of fertility. Female macaques immunized with PH20 truncated into reduced bacterial toxicity related to full-length glycoprotein, also displays significant immune response, suggesting that PH20 can be used for both male and well as female contraceptive vaccines. Sperm egg infusion is a partially facilitated by the pairing of transmembrane sperm protein Izumo with the oocyte receptor Juno. This is what we were talking about in previously also about the junctional protein, the Izumo and the Juno. Loss of functional studies have shown to that knockout of one of these proteins is necessary to inhibit successful gamete fusion and fertilization. In vitro anti-Izumo antibodies were capable of reducing sperm egg fusion events, though no effects on fertility were observed in immunized mice, male or female. Additionally, sperm-specific targets continue to establish for female immunocontraception, though most have not been able to achieve desired fertility reduction in vivo. We have various kind of sperm-specific antigens like the YLP12, the SP56, the P10G, the SACA3, and the FA1 have demonstrated reductions in number of litters and litter size. No complete contraception has completely been observed yet. The recombinant TSA1 and the SP10 were capable of reducing high antibody titers when dosed to female rabbits and macaques respectively, but studied investigation showed that efficacy of these antibodies to prevent pregnancies in vivo have not been performed yet. Surprisingly, fewer studies have been performed in male mammals when it comes to sperm-specific antigens. The synthetic peptide of the sperm flagella protein SPF2 reversibly reduced fertility in 80% in male mice. Additionally, synthetic peptide of the human-specific antigen human sperm antigen HSA N-terminus were optimized in male rabbits and subsequently used to immunize male marmosets. The peptide induced high antibody titers in 7 out of 9 marmosets, but only all but one experienced infertility. Continued research has been established in SAM14, SAM32, ESP, APAP, P4, P3 by 4, and CD14, 46. ESP, especially localized on the surface of sperm between the acrosome and post-acrosomal sheath, has been demonstrated to play an essential role in fertilization through egg penetration. SAM14 and 32 antibodies have shown to inhibit fertilization in vitro, although in vivo studies are required. The Zonas pellucida binding protein, the ZP3, is a glycoprotein responsible for formation of the extracellular matrix coating that protects oocytes and facilitates fertilization, making it a unit instance of the oocyte-specific conceptive target. Genetic studies have been done around the ZP3, necessary for mouse oocyte functioning and fertilization. Side effects were not observed in somatic tissues, therefore, it's one of the best areas to target. Immunization of peptide mimics and recombinant proteins of ZP3 have been under consideration currently. One pilot study in eastern grey kangaroos achieved complete infertility among immunization with recombinant ZP3 in the Freund complete adjuvant. These results suggest that additional factors may be necessary to consider this as a successful immunocontraceptive. Strategies to enhance immunocontraceptive effects include targeting several proteins at once by vaccination with multiple peptides, as well as with direct administration of antibodies. Sperm antigens SP17 achieve limited efficacy alone, but increases in antibody titer were observed when you have a sperm antigen 17 fragment which was combined with GnRH infusion protein. Though not entirely hormonal, this immunocontraceptive technique allowed around 90% of female mice immunized with the fusion protein to experience infertility. Most recently, we have a chimera protein of Izumo, SACA3, and PS20 targeting gamete addition and fusion. This led to complete infertility, reduced around 80% of immunized mice, and remaining 20% experienced a near 50% reduction in the litter size. There are another CD52 which is present only in human and chimpanzee sperm, which can be targeted at monovalent anti-CD52 Ig are expected to elicit the conceptive potential. Lastly, we have the non-surgical alternatives of permanent sterilization. In this, we are targeting, although surgical sterilization is something which is non-hormonal. This we are targeting the sclerosing drugs which are available currently, and we are trying to put it at places which will lead to sterilization. The use of quinacrin platelets for sclerosing human female sterilization have been documented, though poorly supported because of safety concerns. More recently, we have found that the PF, the pilolidocaine form, that have been administered transcervically to cause fallopian sclerosis in non-human primates to prevent pregnancy. But complete sterilization required 5% PF higher doses compared to the one which are approved for clinical venous sclerotherapy, which is 1% PF. So in conclusion, non-hormonal contraceptives can target one or more critical processes which are related to either development of gametes, the transit of sperm, the ovulation of oocytes, or fertilization. Scientific advances continue to identify numerous other proteins and form new immuno challenges which can be affecting the development of sperms or the egg. Many additional approaches were developed to address the gap in non-hormonal contraception, which may promise as in local delivery of new antimicrobials or immunocontraceptives. Continued research and optimization is something which is yet needed. So, with the concept of sharing responsibilities and power in the choice of contraception, it is important to understand perception and outlook, along with delivering newer contraceptives to foster clinical equality and cultural shift in the idea of safe sexual practices and planned health practices. Thank you.
Thank you so much, sir, for the outstanding, I would say. Moving on to the questions now.
Sure, Peter.
So I will mute you. So, with the rise of patient centered care, how do you see the role of non hormonal contraceptive methods evolving in meeting diverse patient preferences, particularly among women seeking hormone free options due to health concerns or side effects?
We all know that can happen because of hormonal contraceptives and the limitations with the weight, the history of malignancy, smoking, and other side effects in the body. So most of the non-hormonal contraceptions are targeted at the molecular level. So as we read about, it will affect the oogenesis, and majority of the contraceptives are the non-hormonal contraceptives that are under research are being targeted for male contraceptives. So that way, even the females don't have to take anything, the responsibility is shared, because if you look into the current data, most of the contraceptives are targeted at women except the condoms which are majorly male centric contraceptives. So, yes, this is how a woman becomes more independent. The partner justice or the judification in a partner relationship also increases, and it protects the woman against the hormonal side effects which can be associated with the currently available contraceptives.
Right, so one more query. In your experience, what factors most influence patient choice when it comes to non-hormonal contraceptives, and how can healthcare providers better address misconceptions or concern related to these options?
Can you repeat the question once again? The mic
Right,
was
so
not being.
in your experience, what factors most influence patient choice when it comes to non-hormonal contraceptives?
So when it comes to non-hormonal contraceptives, the availability and the making sure that there's no hormonal side effect which happens in the body, and also the efficacy. Even if you're using a non-hormonal contraceptive, we have to make sure that the effect is important rather than giving a hormonal benefit, which leads to around 99% contraceptive. If I give a non-hormonal one, which is around 60%, 70%, so obviously the patient will opt for a hormonal contraceptive. So if I have a non-hormonal one which gives me around 90% plus effective rate, then obviously that is the one which the patient will choose for. So that is one thing which becomes very important when you're choosing any contraceptive. The cost of the contraceptive also becomes important, and the ease at which it was used. If you give something which is like a female condom, which is very difficult at times to understand and use, when you have a male condom, it's very easy to use. So the ease at which you can use a contraception, the availability of the contraception, and the efficacy that it can provide will all decide which contraceptive method I have to use and whether I have to prefer a non-hormonal contraceptive over a male available hormonal contraceptive.
Right, sir. Sir, these were the questions for today. So I would like to thank you for such a beautiful presentation. And sir, thank you
Thank,
so much for putting your effort and time for this sharing this invaluable insights. Thank you so much,
thank you,
sir. With that,
Arpita. Thank you, everyone. Bye-bye.
thank you, sir.